So in today's class, we'll be studying about genetics. The scientist that comes in mind when genetics, we study about genetics is G.J. Mendel. That is also named as the father of genetics. He gave three laws: law of dominance, law of segregation, and law of independent assortment. Let's study about the monohybrid cross. What is monohybrid cross? But before just entering into the monohybrid cross, we first need to learn about what are genes, what are alleles. Okay, so genes, they regulate a characteristic in the body. Like humans have 25,000 genes, and each and every gene, they control some biochemical reaction, or they control some kind of a phenotypic characteristic feature of the body. G.J. Mendel was working on garden pea plant, and on the garden pea plant, he found that there are many genes. And out of those genes, let's take the example of height of the pea plant. For height of the pea plant, suppose height. Okay, because it's a monohybrid cross. Monohybrid cross means we are just using one characteristic feature at a time. So genes, that is controlling height. And alleles, these are alternating parts of the genes. Suppose we have allele capital T and also we have allele small t. So the capital T, it is called as the dominant allele, whereas this small t, this is called as recessive allele. Dominant allele and recessive allele. So these alleles, these are alternating characters of a gene, alternating parts. They generally, they are present in together. They are present together always. So genes can be of three different types. Either it can be capital T genes, capital T, or capital T, or it can be small t. The capital T will be called as homozygous dominant gene. This is called as heterozygous gene, and this will be called as homozygous recessive gene. So let's make a cross. First of all, we need parents. Let's see the parents. Suppose I want to make a cross between two parents, male and female, and I'm selecting capital T as my male parent, and I'm crossing it with, let's see, small t. Okay, this is a particular process. This is a case one. Okay, we need to make gametes out of it. So the male plant will have pollens, and because it is homozygous dominant, all different type of gametes that are possible, they must be capital T and capital T in this particular case. For female, it should be small t and small t. Okay, so if we are crossing it, there are various different methods of crossing. This will be called as filial 1 or F1 generation. Okay, there will be 4 progenies, and we can have 4 progenies by using Punnett square method. Okay, I'm taking the male alleles, male genes this side, gametes, and the female gametes this side. And if we are crossing all of them, because gametes will gonna fuse together, Ultimately, male and female gametes will gonna fuse and form the zygotes. Four progenies will be formed in the F1 generation, and all will be capital T and small t respectively. These are the four generations. And if we are going to the next generation, suppose F2 generation, if we are going to the F2 generation, then we can cross these two together. But before that, let's see the phenotype and genotype. Phenotype and genotype. So phenotype refers to the outer characteristics of a plant, of an organism. These four progenies will all look same because these all are heterozygous dominant, right? All tall, we can write this. And for the genotype, also all will be the same. All are tall. Genotype means we need to check that all are tall and all have same alleles, same set of alleles. Capital T small t, capital T small t, capital T small t, capital T small t. These are called as genotype. Suppose if capital T is also present, then we can't write this. We need to write the ratio. In that case, we generally write the ratio. Suppose if I'm making parents again and I'm taking these two, capital T small t and capital T small t, guess what will happen in the gametes? This time, gametes will be capital T and small t and capital T and small t. And if we check the F2 generation, you will see different type of ratios this time. And why it is so? Because suppose this is the male and this is the female. I've written their gametes. This will be capital T, right? This will be capital T small t, capital T small t, and small t small t. And the phenotype will be, because from the exterior characteristics, I am seeing that these three are tall, where this is not. So 3:1. Whereas the genotype, because this tall on the level of genes is different from this tall, right? So we will see that because it is homozygous dominant, these two are heterozygous dominant. They are not similar when it comes to the genotype. So 1:2, 2:1. And the small one, small t, is the homozygous recessive. So this was the whole monohybrid cross. Do let me know if you have any doubts. Thank you.